Let me introduce our first speaker. This is Professor Daniel Olive from the University of Marseille, also scientific founder of Imcheck. Daniel will introduce butyrophilins, their role in gamma delta T cell biology, and the use of anti-butyrophilin antibodies. Daniel?
Thanks for introduction, and I thank as well Paul for having invited me to this meeting. I think it's a really timely meeting. It's a really pretty good timing. Last year, for instance, in PEX in Europe, in Boston, it was a little bit too early. Now it's getting good timing, I would say, just to push a field. I think it's a pretty good idea. So these are my disclosures. As said by Andrea, I'm co-founder of Imcheck Therapeutics and have stocks within Imcheck Therapeutics, of course. So my presentation will be in 4 parts. First one will be a general on gamma delta, mainly Vg9Vd2 and butyrophilins. And I will then allude to other topics, and the end will be the current clinical trial, that is the eviction clinical trial from Imcheck Therapeutics. Since I'm the first, I thought that we need to put this one. I'll let my colleagues who might discuss Delta 1 present something more specific, just to have a general view for a broad audience, I would say. The gamma delta are here, are within the what you call innate-like, meaning in between adaptive, because they have a TCR on the cell surface, but not recognizing peptide MHC, but more metabolic stress for the delta 2. There is a limited receptor diversity, but like innate cells, NK for instance, have a very rapid response and seem to have a more limited memory response. So they seem to be, for a long time, cells are important because of also infectious disease, because, for instance, in mice, a protective role in different cancer models that were found. Our friend Larry Lamb in '96 was the first to demonstrate that there was an increased frequency of the gamma delta T cells post bone marrow transplantation was associated with better survival. So they were prognostic factors. The last set of publication came in 2015 when Gentles published that through transcriptome analysis through different solid tumors and hematological malignancies and cybersort investigation, the population that came first as prognosis factor was gamma delta cells. This was confirmed and extended by Tozzolini and Meraviglia in 2017 in different solid tumors. The cells I will discuss now, the Vg9Vd2, are specific in the sense that they are able to recognize modification metabolism on cells, specifically in tumor cells, by increase in phosphoantigens that is generated by perturbation of mevalonate pathway. Another source of phosphoantigens is coming from mainly intracellular bacteria through the generation through aroma cycles of different phosphorylated compounds that both will activate Vg9Vd2. So it's really something really totally different from T cells and something more related to this type of compound. From this slide, which is really busy, maybe more useful for the panel discussion, except what is in bold, the rest might be used later. Just to indicate within the landscape since the discovery of gamma delta in the early 80s to now, the different ways. The first one that is important was when in my lab we identified BT3, that turned out to be at the end BTN3A, that as a new member of the B7 family. And for years I tried to understand what they were. It took some time till 2012. Basically, we found it in 2009, but it was published in 2012, where we could demonstrate that BT3, now BTN3A, was implicated and was a key regulator of Vg9Vd2 cells and involved in the sensing of phosphoantigens. And later, 2020, two things that are important. The first one is that it was demonstrated between BTN2A1 was one of the ligands of the Vg9 TCR. And Imcheck Therapeutics launched the clinical trial called Eviction using activating BTN3 antibodies. And more recently, what we found in the lab and together with Imcheck is that there were ability to have a partnership between BTN3 and BTN2 in this function in cancer setting, mainly primary tumors as shown. So what are the butyrophilins? The name comes from milk fat globules because initially they were found in milk fat globules in cows. And the first member is called BTN1A1. And after it was found in many mammals. And there are BTN homologs in vertebrates. The family is rather large because what we found is that it's a multigenic family. So far in mammals, we consider that 14 members have been identified so far. The difference is seen here because, for instance, BTN3A1, A2, A3 is present in humans, but not in rodents. By contrast, the skint family was increased in rodents, but there is only one pseudogene in primates, is skint. So it means that there is an evolution that is divergent between rodents and primates in general. And the reason why we have more BTN3 and BTN2, for instance, is not so clear at the present time, but we need to insist on this. In terms of structure, having a look at this molecule, it possesses two types of domain. The first one, the extracellular domain with IgV and IgC, like B7, is a way Peter Linsley from BMS described it in the early 90s. And what was puzzling is that within the intracellular domain, there is a B30.2 domain. So this domain is not so familiar for people doing immunology, but anyway, it's important because it was described to be a sense of innate immunity through mainly cytosolic molecules, like TRIM5 alpha, for instance, involve the prior decapsidation of HIV, or the pyrimidine that is involved in inflammation. And the reason why it was found this way was surprising. In a nutshell, because I will not present all what has been done during the last 10 years regarding BTN3, it just, this molecule, and this was one of the problems initially for me to understand the function, that the molecule is expressed on tumor cells from diverse origin, as well as normal cells, including immune cells. Vg9Vd2 can be recruited like they are found, for instance, in TB infection. They can recruit in some tumors. What we could find is that the regulation of BTN3 depends in tumors on hypoxia and nutrient deprivation, as well as inflammatory cytokines. And in most of the tumors we have tested so far, recognition of tumor depends on BTN3A. I will not discuss today because we don't have time, in general, the different subtypes of Vg9Vd2 present in cancer patients. Rapidly, in terms of function, is shown here. This is a classical assay where you use Vg9Vd2 and you incubate them with lymphoma cell dowdy. What you get here is a degranulation. This is the CD107 assay. The activating antibody called 20.1 induces a very massive degranulation. By contrast, if you use the inhibitory antibody antagonist, you totally abolish this effect. What is surprising, and took some time to fully understand it as well, was the way the antibody were acting, because the molecule is present on the target and the effector. One way to try to decipher this was to do pre-incubation experiments and to add the anti-activating antibody 20.1 to the effector cells, wash, and then put them on the target. Nothing happens. By contrast, if you put the antibody, incubate it with a target, wash, and now come back with your gamma delta, you have a nice kill. So surprisingly, it acts mainly, if not only, on the tumor cells, that is the cells that will express the virtual antigens. The second part is more biological. Try to understand the way it works in general, and the surprise came last year when it was found that BTN2A1 could be a partner of BTN3. Just to come back on the chromosome, so on chromosome 6, you have a lot of members of the butyrophilin and BTNL family, BTN3, BTN2, BTN1, MOG, as well as BTNL2. And in close vicinity to BTN3, BTN2 is present. With 3 members, BTN2A3 is a pseudogene, whereas in mice, there is only BTN2A2 that is present. So what was done, published this year, was to understand the way it worked and try to reconstitute the system, because I told you there is no function of Vg9Vd2 in rodents for 2 reasons. The first one, there is no Vg9Vd2 in rodents. So with the second problem, our colleague Thomas Herman devised a model of myosin hybridoma transduced with Vg9Vd2. And what we did was to transduce myosin cells with BTN3A, BTN2A1 or A2, or both. And the only condition where we found the ability to empower the myosin hybridoma of response were when we transduce BTN2A1 and BTN3A1, not BTN2A2. So it means that we can reconstitute the system this way, and this is a final demonstration of the 2 molecules are critical, and they act as heterodimers. Where we are at this stage of recognition by Vg9Vd2 is that gamma-9 is able to bind, among others, BTN2A1. Rigo and Al propose that there might be another protein present in cancer cells that might recognize delta-2. We don't know yet. And BTN3A1 was proposed to recognize another ligand on the gamma delta cells. Among the possibilities, Payne and al. Recently described last year that CD45 might be one of the partners. It might be, that's right. The only point is that CD45 has a broader tissue distribution, of course, because this is a marker of leukocyte, but it has to be considered because it's a phosphatase, of course. So at this stage of a conclusion for this first part that is more biologic, I would say, what we can say is that the co-expression of BTN2A1 and BTN3A1 is mandatory for the function of Vg9Vd2, cytotoxicity as well as cytokine protection. But BTN2A2, surprisingly, doesn't permit this function. What is the function of BTN2A2 is so far not known. We find them co-expressed in most tumors so far, in leukemias, and there's still open questions that are what is the range of different ligands that permit to get this specificity of function of Vg9Vd2 against the tumor expressing BTN2A1 and BTN3. So the step after was to go towards more biology on one end, at the same time to go to clinical trial. is the objective of discussion from today, of course. And I will take 3 topics for what we call a preclinical. The first one, I take an example, this is not yet published, on a thing done in leukemias. And what you can see when you are doing in an autologous setting, because the critical question when you want to work in patients is specifically when you consider using antibodies to have gamma delta cells that are able to work. And this did this, for instance, in leukemias, in ALL, so this B and T cell ALL. And you can do using autologous Vγ9Vd2. You have a very good killing, but only if you activate BTN3A activating antibody. Meaning that although these cells are poorly recognized by gamma delta, using the antibody, you have a very massive killing apoptosis of these cells. And it's shown as well by themselves, leukemic cell doesn't permit to have degranulation and cytokine production, but as soon as you add the activating antibody, it works. So it means that in this setting, autologous, you can get function of cells. The second, although for me, in part artificial because it relies on immunodeficient mice, since we don't have an autolog, so the only way to have a proof of principle in a murine model, in addition to in vitro experiment, to do in vivo using a PDX of AML in this case. And in this setting, what we are doing, because the immunodeficient mice doesn't have gamma delta, you infuse gamma delta cells from allogenic donors, and they control in part the leukemic cells in the blood and in the bone marrow. This control is increased if you add the activating antibody because you have no more cells in the blood and a very major decrease in the bone marrow. The last issue was a problem safety. For me, it was a concern since when I launched the company in 2015, it was the only way to be fully convinced that a molecule with a broad spectrum of expression could be used and could be targeted. And what was done was to use the phaneromalgus monkey as NHP. And it turned out to be a nice model because there's expression of BTN3A that the antibody binds well to the cells and different targets. The antibody was able to have a good in vitro activity on both human and cyno Vγ9Vd2. And in vivo, after infusion of 3 different doses indicated here, there was a very nice target occupancy occurring rapidly after. There was a nice activation of the Vγ9Vd2, shown here by C69 induction. No dose-limiting effect in the animal using these doses, and no CRS, because it was one of the major problems, whether you could get a cytokine release syndrome in these monkeys, unknown, appeared. And finally, in addition to absence of safety concerns, there was a very nice effect on the Vγ9Vd2 that was specific because it was not found on B cells, for instance, of T cells, with a rapid decrease on Vγ9Vd2 in the peripheral blood of cyno to go, we assume, to the tissue. With this green light, we could then go to the clinical trial. And so where we are regarding clinical trial, just to explain at which stage? So the clinical trials that was launched by Paul Frona, the CMO of Imcheck Therapeutics, started in February 2020. You know, that is an interesting date because it was just in France, just before the lockdown. But anyway, the investigation could be done, and we have to thank all the centers who could be able to run the different trials in France, Belgium, Germany, Spain, UK, and US. Anyway, so the dose escalation part was presented as ESMO and SITC recently. On one hand, the group A is a monotherapy with a basket clinical trial with different solid tumors. So far, 32 have been enrolled. Second, methodological group data will be presented today. And third is a combination with anti-PD-1. In this case, what we used was the Keytruda in patients. And this part, this one has been completed. 1, 2 is still in progress. Then the clinical trial will expand to have expansion cohorts in selected patients, and this will be the objective next year. The first and really important information in the 32 patients throughout doses, that is from 20 micrograms till 200 milligrams with different tumors. There was no dose-limiting toxicity, no safety concerns, which was, of course, important, again, due to the distribution of a molecule, and there's no cytokine release syndrome that could be identified. I don't show the data, but at the same time, the saturation of antibody, the PK was good, as expected and predicted from initial study in NHP. And there was a production, dose-dependent production of cytokines in patients. One interesting piece of information came for the translational studies in this patient. Based on 32 patients, I will show 2 that we could explore more in details. We didn't have all sample for all the patients at baseline and after enrollment. I take an example for immune desert tumor. This is gastric cancer. So nothing, meaning no T cells, no CD8, no gamma delta. But after infusion, there is an increased gamma delta shown here. This is the digital pathology indicated here. T cell as well as CD8. This is the first example. So first extreme immune desert. Another example, a more classical ovarian cancer with immunofiltrated tumors that you can find here with gamma delta, CD3 and CD8. Anyway, after infusion, you have a further increase in the cells, the CD8, as well as the gamma delta. So phase 1 provided this information, as well as additional things that I would just only describe without showing the data. I'm sorry, but I can show the data if needed during the roundtable. So as soon as the antibody is infused, in a few minutes, the cells are leaving the blood and moving to the periphery. So there is an intense trafficking out of the Vg9Vd2 in the blood. We can find after really early time points, 30 minutes, production of cytokine like interferon gamma and TNF-alpha to not very high levels, but that is significant in comparison to before. And in some patients, not all, we can detect effects on NK and CD8 meaning at later time points, the cells are trafficking out of circulation as well. And we find them in tumors. So it looked like there is at the same time that the specific effect of the gamma delta 2, that is very early, a secondary effect on other populations. So to conclude this part, so with a good safety and tolerability through very wide range of doses, the intention to treat efficacy is best was stable disease in 6 out of 32 patients. And so it means that we can consider to continue of our expansion phase and combination. So combination, the idea is rather straightforward because PD-1 is also molecularly important on gamma delta cell biology and is one of the gold standard in different cancer. So for this purpose, what we did was to use patients with at a flat dose of 200 milligrams of pembro, Keytruda, and 3 doses. So far, 8 patients are enrolled with 700 micrograms, 2 milligrams, and 7 milligrams. So the trial, the accrual is continues at present time. Regarding target occupancy, this is shown here, there is no modification in comparison to monotherapy, meaning there is no perturbation when we are the anti-PD-1 at the same time. Second, there is no increase in cytokine production or tox, because of a combination of both could have been a problem. And what we can detect for gamma delta, again, rapid decrease on the gamma delta in the periphery. In some patients, there is a decrease in NK and maybe a little bit of CD8, but it's something that is less convincing. But so far, we don't have enough patients in comparison to 32 of a previous study. If we have a look at the spider plot and we analyze the 8 first patients that could be evaluated, again, the study continues. What we notice first is that we have an objective response rate of 2 out of 8, is not a lot, of course, it's 25%. And if you consider the disease control rate is 5 out of 8, meaning 72, which is rather interesting, I would say, based on the fact, because if you take the example of what is indicated in green at 2 milligrams, correspond to a patient with melanoma who is refractory to nivolumab plus ipilimumab, meaning the double therapy, so highly refractory. But anyway, there is an effect, and you can be shown here, even more striking, if you have a look at the very same patient who had asymptomatic brain metastasis. You can see here, after 1 infusion, there's a decrease of the MET that is no more detected after week 27. And the effect on the liver MET is more classical, I would say, of what we expect of classical immunotherapy like CTLA-4 or PD-1, because you have a pseudo-progression comparison of baseline, pseudo-progression, then a decrease in the tumor. So to conclude this part, before the final present conclusions, is that the trial is still ongoing, and we're waiting to have more patients with more doses of HTO1 and pembro. There are preliminary signs of tumor regression, have even low doses, which is a surprising 7 micrograms, which is not a lot, and 2 milligrams. so which is a surprise. So we may think that the recruitment of cells may modify, remodel the tumor immune microenvironment and increase the effect of anti-PD-1. This is a guess. It has to be demonstrated, of course, and we need to expand this study. So far, to conclude, the B7 family members, BTN and BTNL family, provide unexpected function irregularity in innate cells. This permits to get better understanding of the function of Vg9Vd2 and maybe other population. We have seen the effect with anti-PD-1 as well. And one of the lessons is that despite broad distribution, we could target this type of molecule, which is unexpected, and activate using the antibody and in a safe manner, and have something that is specific for the Vg9Vd2 in both model NHP, cyno, and patient. We need so far to have more information regarding discovery first, understand better the biology and function of the cells in terms of expression function and molecular interactions. And among the urgent things to understand is molecular mechanisms that might explain and demonstrate fully the interaction between both pathways. So it's a long-standing effort since 2004, I would say, from different investigators and students, as well as clinical staff, and for the eviction trial in Imcheck therapeutics with different people. And of course, the patient will participate to this study, as well as the different centers throughout the world who are still participating in it. I will stop here and we'll take the question.